Welcome to the Axial Podcast. Axial is an early-stage investment firm based in San Francisco. We partner with great founders and inventors investing in early-stage life science companies often when they are no more than an idea. Axial is fanatical about helping the rare inventor who is compelled to build their own enduring business.
Okay, Luke, we've been talking for like 30 minutes. Thanks for joining this podcast. We're going to do this like every 2 years. I'm a huge fan of your work. Maybe for people listening, you give a brief introduction about yourself and the work you do, and then we can just start the conversation.
That sounds great.
Also, we're doing this in person. So thank you to Playground and Jory Bell for hosting us.
Yeah, no, it's great. I was just at a CZI workshop just up in Redwood City. They're doing a lot of cool stuff in imaging. So that's why we get to meet in person. But I'm Luke Lavis. I'm right now a senior group leader and head of molecular tools and imaging at Janelia Research Campus. And my lab makes small molecule fluorescent dyes, and I've been doing this for almost 25 years.
Cool. Well, you're definitely like the key person for a lot of like super resolution imaging. Maybe the key person in Janelia in general, and the key person for a lot of people's research when they want to image anything. Maybe we can like start off from the beginning of how did you get to chemistry and imaging? Where'd you grow up? How did you get involved in science? What was your first job like? How did you decide to, how did you decide to like even try to become a professional scientist versus like probably a lot of other options and interests you had?
Yeah, so I grew up in Southern Oregon. My parents were both hippies. My mom grew up in Orange County. My dad grew up in Santa Cruz. They moved up to Southern Oregon in the 70s. My mom lived in a commune, and they met, married, and we kind of lived out in the sticks. And I enjoyed building stuff. I enjoyed hiking around. I didn't necessarily think of chemistry as a first career when I was little, but I was good at math. I was good at science, and so when it came to finish high school, I almost didn't go to college, but decided to go to Oregon State University, a first generation college student. And then at Oregon State, I kind of bounced around. I was an anthropology major for a little while. I was an English major, and then chemistry really resonated. So I went back. I had the opportunity to work in a lab run by James White, who sadly passed away a few years ago. And he was a natural products total synthesis chemist. And so he gave me a bench. He treated me like a graduate student. I was supposed to work 40 hours a week. I was taking full classes. So there, Saturday mornings, he would walk through the lab and make sure everyone was there. It was kind of old school organic chemistry lab. I learned a ton of chemistry there, and I learned that I really didn't like natural products total synthesis. Yeah. So when it came time to take the next step, I wasn't sure. I loved chemistry, but I didn't want to do natural products total synthesis. And so I found the one job you could get as a chemist in Oregon, and that was at this funny little company called Microscopy in Oregon.
My mom went to Humboldt State, and she has like some hippie values as well. But I'm not a hippie, even though I try to act as once in a while. Like, are you a hippie, or do you have some values that have those values kind of steeped into you? And then how did that influence you going to college? Like in terms of, when you got to college, were you very oriented towards something humanities or something more like, I don't know how to characterize this the best, but like something more like free and open versus something like chemistry, which is very like very old, total synthesis. I don't know who James, I'm not familiar exactly who James White is, but I'm assuming he's, a lot of chemists I know wear suits. They're not wearing, Grateful Dead shirts. And so.
Yeah, that's a great question. So my original plan was I wanted to go to medical school, and then I wanted to be a missionary doctor in the South Pacific. That was my dream. So, this chemistry thing came in, and I thought, oh, I'm good at this. And I wasn't sure, okay, go to grad school? Do I go to medical school? And that's also why I was doing some of these other things. I thought, oh, anthropology really interests me, and it would be distinctive to be an anthropology major with a strong science background and then go to medical school.
Interesting. I'm trying to make sure that, like, I did chemistry because I want to make LSD or something.
No.
I gotta do chemistry. that'd be a cooler story, but not reality. But same thing for me. I wanted to join Peace Corps actually.
Yeah.
And then I realized, I don't know, for me personally, I don't want to go to like Zimbabwe or like Colombia. But how did you realize you're good at chemistry? Did somebody tell you, or did you synthesize a molecule first try? Like,
I don't know. There was just something about it. It resonated with me. I think I'm a very visual learner, and so the beauty of chemistry is it's all pictures, right? And so there's a strategy building molecules, but in the end, there's these pictures of molecules, and you say, I'll make the thing. I have these tools available, and so you can do it. And the other cool thing about chemistry is it's like a hands-on science, right? It's like cooking in many ways. It's hard to automate, although people are having some success automating it. But in the end, it's a person at a hood, finally manipulating these solutions and getting things to work, right? There's joy in making a new molecule.
Okay, so you're in Oregon State in the lab. Was this like your freshman?
Yeah, like sophomore.
Yeah,
you're on.
You took some chemistry courses, and then you joined the lab. I joined the lab around that same time. And I was amazed, like, they just let me do anything?
Yeah.
Like, what? Like, I can just do anything I want in this lab and no one's going to really tell me what to do? And that kind of got me hooked a little bit. Any kind of horror stories? Did you break an expensive glass? Because I remember I burnt like a bench once. Because like I put the Bunsen burner underneath.
Oh, okay.
And they got really mad at me.
Yeah.
Like, any stories, like, the first, like, year, 6 months, like,
the first reaction that I did ever, independently, it was this production using Perlman catalyst, which is palladium hydroxide, and I didn't know anything. I was actually making a natural product found in pine trees, a little side project that Jim had funding for, and it was meant to be a standard for GC for gas chromatography with someone in the forestry department.
Yeah.
It's a natural product found in pine tree bark, and so I made this thing, and then I was taking off protective group. I didn't know. It's like, okay, I asked him, what do I do now?
Yeah.
Filter it. So I filtered it. I realized that this palladium hydroxide reacts with air. So then I picked up the filter, and it was going hot, right? So it's like, crap. You know what I mean? Like dump it in the sink or something, right? So I learned, I had a bench. I was treated independently. Jim didn't take very many undergrads in his career. He only took 6 or 7. So it's really an honor to be part of that group.
Cool. Yeah, same thing. I worked with Gear Ruffkin. It was only postdocs and undergrads. And it was like me and 12 postdocs, and I could do whatever I want. It was exhilarating, but then terrifying.
Yeah.
It didn't break anything. Okay, so you're in Oregon State. Any kind of like you're doing research in the lab, like how did you get the leap to become a professional sidentist? Something I experienced too, like I had science friends, I guess, people who studied engineering, chemistry growing up, but I didn't really grow up like a professor, like a professional scientist who made a living publishing papers and doing work. How did you finally realize, hey, maybe it's something I can do? Like, or did it happen in college or maybe later on? But
I think I'll be honest. I think part of it after my undergrad work is I wasn't quite sure if I was good enough, and so that was one of the reasons to go to industry. One of the things that I do now in my lab and for this effort that we call open chemistry, where we share a lot of the reagents, is I want to give other folks coming out of undergrad, before they go to grad school, an opportunity to work in the lab. I think in general, we don't have a lot of these so-called post-bac programs. Hughes is starting a new one, which is great. I'm part of the working group there. But, I think those types of opportunities do do a few things. They teach people, yes, you can work in the lab, right? You are a good scientist. You're able to be productive, and also shows folks that it's fun. It's a job, right? It's go into the lab and make something new every day. I mean, that's pretty cool to do.
Yeah, absolutely. I remember Rich Losick had a program with HHMI where we get like students from like maybe universities that aren't like where a lot of opportunities are, and then get them to like a Janelia farm, a Yale or something. And
yeah, so that's the goal with this new program, and then I've been kind of doing it, on the side, I guess, over the years. Several students have come through the lab, and then most of them have gone on to graduate school.
Cool. Okay, so you're at Oregon State, and you joined New Industry, and it's amazing to hear. Oh, maybe I wasn't good enough, and now you look at you, you're like the best person in imaging guys. It's crazy. That kind of tells you that big journey is a little unique, and there's no like standard path to being the best. And so you're at Oregon State. Why did you join molecular probes? Was it just because it was down the street, or a friend worked there, or
it was? I mean, it was partially because I love Oregon. Okay, and my wife, my awesome wife, also from Oregon.
Cool.
And so I just never ever wanted to leave. And so I'm like, okay, I want to find a job here, and I'll figure it out, right? So I walked in. I think I mentioned Jim, I only took a few undergraduates. Well, one other undergraduate was working at probes. Former undergraduate was working at probes. This awesome person, Kyle Gee, and so I think that got me a little bit in the mix. And so I walked in, and I remember during my interview sitting with Kyle, and I asked him, how hard is this? How hard are these things to make? How much do you care about stereochemistry, which is kind of the bread and butter of natural product photosynthesis? He said, "Oh no, we're not trying to make fancy molecules. We're trying to make useful molecules." And that's really what sold me on this idea of molecular probes, and that's why I wanted to go there. Cool. And I think that one thing, making useful molecules versus cute, interesting, complex molecules is something that I really go after still to this day. Because it's all about the utility, not just about how sexy they look.
Okay, so this is probably a very formative time in your life where you begin to establish like what you actually want to work on. So like, what did you do on molecular probes? Was it just the same thing you do now, or maybe related, or just?
Yeah. So my first project there, they had just identified all the Alexa fluor dyes. So this was in 2000.
Yeah.
And so my first project is you're going to make a gram of Alexa fluor 633, one of these Alexa fluor dyes. And so give me the synthesis. They'd already identified this as a candidate, so I just had to go through and do this. And the other funny thing about dye chemistry and working in industry is you don't necessarily care about the yield. The rules that I learned in an academic natural products lab, where it's got to be 80% yield or try again. I did my first reaction. I got 65% yield, and I felt really bad. And then I told my boss, and he's like, "Oh, that's the best anyone's ever done on that." So I realized, this is kind of fun, because it's not about calculating how much you get at the end. These dyes are so useful, so valuable, and you don't need that much. So it's just about making them at all.
Cool. Maybe we'll give a primer on imaging dyes later on. I think the story's more important. So you molecular probes for a few years, and then you moved to molecular devices. Which unrelated, I think.
Well,
or are they related?
So what happened was, I was at probes again. I love Oregon, but I worked for my supervisor, this guy Zenji Wu. Now he goes by Jack D. Wu. He owns AET BioQuest down in Sunnyvale. Cool. Really fantastic mentor and teacher. And so he left probes and went down to molecular devices. And so he got there and he recruited me down. And so I was only at probes for about a year, and then moved to down Silicon Valley, Sunnyvale, kind of right around the .com bust, which was an interesting time to be in this area. And and then just started there. The Molecular Devices was a company that made plate readers and other equipment for high throughput screening, and they had just started a reagents business. So there were 3 directors, and under each director there were 3 people. So it was a team of 12 in cell biology, biochemistry, and organic chemistry. And a year ago we had, 10 million in consumable revenue. So we built up these kits rather quickly, and we had nice facilities. It was a really fun time because then I started interfacing with engineers who were building equipment, and we were trying to build kits that would work well with this installed base of instrumentation.
Yeah, Molecular Device was one of the best instrument companies that were early 2000s.
Yeah.
So your Molecular Devices, ahead of the time was like, hey, maybe I should start my own company, or maybe I should just get promoted and become a VP. But then you go to Wisconsin for grad school.
Yeah.
How did you, like, at the time you had all the success? You have a wife. Is it? You have a wife, so maybe house, a kid. You want to, you know what I mean? But then you decided to go to grad school. Was that part of the plan, or is it just like kind of just came up and you said, you know what, I really want to work on the scientific problem. Let me go back to academia.
Yeah. So again, this was sort of it was supposed to be a holding pattern for me, right? Do I go to medical school? Do I go to grad school? The other issue, which maybe it's different now, but without a PhD in R&D, it's hard, right? I remember one of the other directors told me, the PhD is a union card. It allows you to get other jobs. I still think that's true, although there are exceptions. And I'd been brought in to Molecular Devices at basically the highest level, Associate 4 or whatever, that I could have as a bachelor's degree. And so I had to make the decision, right? Either I could go into sales or customer support, I guess, or there were other avenues to put it, not within R&D. And I think at that point I realized I really like this, solving problems, research and development, doing nerdy science. I guess maybe I figured out I was a real nerd and I should keep doing it.
Interesting. How was it like to work at, so maybe one quick question is how did you have to dress differently when you work at
yeah.
I mean, I think kind of just wear a suit and tie, or was that just
Still California.
So okay, a little some of my impressions of being a scientist versus being a professional, or it just there's things that you have to pick up about how to present stuff and how to sometimes in science it's you're so much in the weed about the technical details, sometimes you lose why it's important. And sometimes what I've realized, not being in academia, you get the better skill set you get how to value that science. And so do you think having those experiences or high school helped you do things that were useful, not so much
no, that's a great question. So I guess I can think of 2 things that I gleaned during my time, especially at Molecular Devices. So Molecular Probes was this fun, privately owned company. And so that was, run by essentially one person, Dick Hodlin, the founder, along with his wife. And so that was interesting because, their mantra was, hey, if we're making money on something, let's keep selling it. So they had this list of thousands of compounds, and it wasn't the best business model, but it was great for the community. I bring that with me now. Let's make molecules that are useful, accessible, even if they're not the biggest sellers. So that was something I learned from Dick, and I really appreciate that. At Molecular Devices, especially, something that a lot of graduate students don't come in with is this idea to fail early and often, right? Which I think is mantra in the business world. And so I learned that at Molecular Devices, right? And so, you try something, and if it doesn't work, move on. And then, sometimes someone else is going to try that. They're going to get it to work, and you're going to kind of be quasi-scooped, and you have to learn just to be okay with that. right? Because at the time, I made this decision. I trusted my gut. I killed it. I'm going to go on to this other project. And so that's something I learned. And then, we were a small team. And so I had to, I was 22, 23 years old. I had to give presentations to the CEO and other things. I remember being talked about the reagent kits that we had developed, right? And so I learned a little bit of how to present things, how to sell things.
I think try to present science and research to people who know nothing and are trying to make money off it. You have to really distill what's valuable. It's actually hard initially.
The nice thing about probes is usually the cost of making things after they aren't as expensive, but the ultimate cost of things is cheap, right? So we were selling a kit for I think $1,500, and the bill of materials was $5. So the CFO, that was the message the CFO understood.
Okay, so you go to Wisconsin for grad school.
Yeah.
I'm always jealous of the people. Something I wish I should have done. Probably should have done 3 years between college and grad school, and then just get experiences and then make the best decision.
Yeah.
Don't go to college and grad school unless you're a genius or some sort of savant. Otherwise, even if Luke Lavis has to get a job and before grad school, I think everybody should. But why Wisconsin? So you're in Sunnyvale.
Yeah.
Why not? Why not? Wisconsin.
I think there was still a little bit of imposter syndrome here, and so I wasn't quite sure. I should have, maybe, I don't regret anything. But I could have. I now know I could have stayed. But so we were a little tired of the bustle of the Bay Area. We lived in Sunnyvale, so I had an easy commute. My wife worked. She's a dietitian. She worked at Menlo Park, just downtown, at this assisted care facility. And so, our life was get up really early. She would take Caltrain up, and I would drive to work. And and it was with 2 kids from Oregon, it was a difference, right? It was a different life. And so we thought, let's go somewhere crazy. And so we thought, okay, where's a crazy place? And it's I had been to Madison before.
Cool.
On a chemistry conference trip, and so I'm like, oh, this place is amazing.
Cool.
And so we decided let's live somewhere kind of different.
I've been to Ann Arbor for grad school, and this place is too cold.
Yeah.
And I guess Madison's not as cold as Ann Arbor most of the time.
There's but it can get pretty cold.
Okay, so you go to Wisconsin. Did you kind of know exactly what you want to work on?
No.
Or you just show up and say, I'm doing rotations, I'm going to explore and try to. Did you think about trying to continue to work on probes? Or were you just open-minded about anything? Hey, maybe I'll do something in stem cells or.
Yeah, it was the chemistry department, and at the time, they had a great chemical biology program.
Yeah.
Now my PhD advisor and his wife, Laura Kiesling, PhD advisor was Ron Rains. They've now moved to MIT. So, so that it's still a great community there, but I think some of the people have moved on, and I was sort of pegged as the fluorophore nerd, in my class from the beginning. So I would always ask questions about fluorescence. So I kind of wanted to keep doing this, and fortunately, Ron Ron's an incredibly creative scientist, and he had come up with this interesting strategy to make fluorogenic probes that would turn on in response to enzyme. And so I wanted to come, and at the time, Ron didn't know my name, but he would call me the probes guy. And so he said, sure, yeah, you can join my lab, probes guy. And so in the lab, we all had nicknames. So my name was Prober. So we would all call each other by our. So it's kind of a fun lab.
I did a podcast with Peter Turnbaugh. I had to publish it, but his old mentor called him the professor. Hey, professor, just because just to kind of give him the encouragement, he can do it. Okay, so you kind of doing graduate research, working on probes, enzymes. Maybe you can describe your project, but more in the context of how did you think about, like, I think when you're in graduate school, you have a higher level of independence, not only in terms of like maybe undergrad, let you do anything, but then you're often being told what to work on maybe.
Yeah.
But in grad school, you're kind of now expected to formulate your own hypotheses, maybe establish a project. How did that come easy to you, or how did you get those skills, like for your project? How did you like drive forward? Because that's kind of can definitely be like the biggest challenge. It can be a lonely existence sometimes as a grad student, where like often it's just the mentor, to be honest.
Yeah.
It's probably the mentor that helps.
Yeah, I mean, in industry, we had a bit of leeway.
Yeah.
So at the time, the big product, which is actually still a product, are these calcium screening kits, right? So we would make it. We were actually making one of the components, and then the company was moving money around to reimburse us for that time. It only took a few days, a month to do this, and so the rest of the time that would pay for all of our salaries and reagents and everything, so we could really just do R&D. And so that was fun, and that gave me a little bit of freedom to explore. So I learned that from industry too, and that's something I try and do for the technicians in my lab. Is they're not just doing what they're told; they eventually get their independent project and get to struggle with it and try new things.
Yeah, this is all build up to the culture we have now in Janelia, where you built to like a really like a center of excellence, and you'd be able to recruit and retain Jonathan Grimm, Brian English, a bunch of great people to like just continue to pioneer imaging dyes and probes. And so it's kind of it's a consequence of these experiences, though. And so you're in grad school. Any kind of like low moments you realize? Did you ever? Okay, everybody has that moment where like maybe it's year one, maybe it's year six. We're like, I don't want to do this anymore. Did you have that moment, or maybe not that extreme, but did you have do you have like an existential moment where you're like, maybe I should go back to industry?
It was actually the opposite. So we had a little workshop for potential entrepreneurs in science at Wisconsin. So I got invited to this, and I mean, nothing against industry or VC or anything like that, but at that point, I really realized I was a nerd and I didn't know science, right? It was like, they were like, oh, this is great. You can start companies or do whatever, be in this world. And I realized, okay, I'm gonna do the science thing. I think.
Cool. Okay. So kind of, what was the thing that turned you off? Was it just the clothing? Was it the lack of curiosity? Sometimes, I think about VC and startups are really fun because you can actually make a direct impact on people, but then the other end is like it's hard. You can't explore as much. That's the trade-off.
And again, this is something I learned in industry, and this is a constant struggle with me. Is scientists, you have to tell a story in the form of a paper, right? But we can often make tools that work, that are really useful, but then we still have to publish a paper. And there's a lot of stuff that goes into a paper that isn't necessarily what you need to get a tool out there. And so sometimes I kind of yearn for the industry days where it's just like, oh, we made this thing, we tested it, it works. We could just, start sending it out and, forget about a paper. But the flip side of that is, sometimes you want to follow up on something. Like, I think this is interesting, and there's no market, there's nothing that it's like, well, no, we got to move on to this other thing because this is more important for the strategically for the company. And actually, one of my projects in grad school stemmed from something I had done in industry, and it was something I never followed up on, but I could adapt it for this system that we developed in grad school.
Cool. Okay, what was your order? So what was the project?
So Ron has a little company on this, but he's a trained enzymologist. I learned, sort of the importance of precision and just being grounded in physical organic chemistry principles from him. And he worked on these ribonucleases, very common proteins that are found everywhere. And he discovered variants of these that would kill cancer cells selectively. So we have this protein in our cytosol. Its sole purpose is to bind any stray ribonucleases that would get in. And so he developed variants that would evade the ribonuclease inhibitor protein, and it seemed to selectively kill cancer cells. So we started get a little startup on this, and what we wanted to do is actually visualize the protein getting inside the cell and then track it. Where did it go? What were the kinetics of it? Escape, it would get endocytosis and escape the endosome. And so we wanted to figure out, trace the path of this protein in cells. And so we had developed a strategy where we had a fluorescent dye. It would be quiescent, dark, until it got internalized. And then we had other probes that would stay dark even after internalization, but then only glow when it escaped the endosome, the cytosol. So that was my job.
So like he has this kind of maybe overarching project. You got there. Did you totally revamp the whole work? Because you're the probes guy. Like, did you just enable things that like he couldn't even imagine in terms of like imaging, like trafficking?
Ron probably doesn't remember this, but he presented this project. He said, okay, we have this probe scaffold that a postdoc had worked on, and then he said, okay, your job is to get this thing working on the protein in a cellular experiment. And so, like, if you do this, we have to give you your PhD. and so I said, it's kind of easy. And so we had an idea, again, stemming from second industry. And so 8 months later, okay, I think we got it. And then he's like, well, I can't give you your PhD, so just do whatever the hell you want for 2 years. So that's what I did.
That's pretty sweet. Okay.
Yeah. I mean, it took a little while to like flesh out the paper.
Yeah, but that's good. Yeah. Just do things that are kind of you're really good at, and it comes easy, and then you begin to like. So at Wisconsin, you had this maybe period of time where you kind of knew you get your PhD, a few years to kind of explore. That seems pretty formative, where you can kind of explore ideas, read papers, go to talks.
Yeah.
Like, what was in your mind at the time? Like, was it like what kind of problems did you? Maybe a better question is around this time, or maybe earlier or later. When in your head did you start solidifying? These are the problems I want to work on. These are the things I'm really excited about. Was it around this time, or maybe?
One of the things that I struggled with in grad school was, this is going back to the chemistry roots, even though I was making probes and interfacing with cell biologists and biochemists in the lab, the struggle is really making these molecules, right? And we were making the simplest rhodamine, this dye called rhodamine 110. It's got it's got nothing on it, right? It's like the simplest dye. It's in the original rhodamine patent from 1887. And, here it was, 21st century, and I was making it the same way that Marie Sarasol, this German chemist at BASF in 1887, was making it. And this involves boiling things in sulfuric acid. And I'd come home, I'd have the holes in my pants, and I was dripping sulfuric acid. It was quite nasty. So I think it was that point when I realized, okay, if we want to break this field open, we got to stop using this 19th century chemistry. This has really stymied the field.
That makes a lot of sense. It just seems like, you're in college, you like chemistry, you don't like this full stuff, natural product stuff. It's kind of really complicated and kind of hardcore culture. A lot of the natural product people I know are like hardcore chemists, like borderline abusive. Some of them are, not all of them, but borderline abusive. I have some friends who.
The culture is it's like 2 hours on Sunday to do your laundry type.
Yes, I don't want to name names, but I have some professors at Harvard I do, and they're very, very hardcore. And so then you get into probes because you join this company, and you work in industry, and then you go to grad school and be the probes guy, and then you just do something real easy, get a PhD. And I'm just trying to like understand, like it seems like you're just really good at chemistry. It seems like joining molecular probes was pretty low.
That was the thing. I mean, I never looks back, right?
It seems like that was like the key, like just having a friend from I've made be in that company as well.
Mhm.
You just join, like, that seems to be like the key thing where it's just like, whoa, okay, I'm gonna just join this company, and then that sets you up
Yeah.
for a very long time. Okay, so you're near the end of your PhD. How did you decide Janelia Farm? The Janelia Farm is a pretty sweet place. It's Chevy Chase, Maryland, or
No. So
it's
yeah,
somewhere else.
Janelia is part of HHMI.
Yeah.
So the Howard Hughes Medical Institute. HHMI is one of the largest philanthropies funding biomedical science in the country. Started by Howard Hughes in 1953, and back in early 2000 or so, they decided to start their own research campus. So before that, they were funding investigators all over the country, but they had no lab space of their own, really. And so they decided to do this, and it's actually in Northern Virginia. So it's about 35 miles west of DC.
I'm sure the point is perfect for you.
Yeah.
Like, think about, like, hey, go to Oregon in a cabin, or not a cabin maybe, but like something equivalent, and you're like, I want to. Janelia Farm is like, it has to be like if I think about for Luke, like probably the best place for you to research.
Yeah.
Kind of fits you, it meets all your criteria.
Yeah.
It's like your dream job, essentially. Yeah. Is Erin Roche still president of HHMI?
Yeah.
She got her PhD in 2 years.
Oh yeah.
At MIT. She brags about that.
I know.
So maybe that could have been
there
you.
It is off scale. Yeah.
That could have been you. 2 years at Wisconsin. Okay, so it's like 2008, I think.
Yeah.
7, 8.
Yeah. So at that point, I've been in the lab 4 years.
Yeah.
And to be honest, I was kind of having a issue finding a postdoc. I was sort of an odd candidate.
Really?
Yeah.
What?
Yeah.
What?
So it was a hard time. And then kind of on a whim, I applied to this new place. This place called Janelia Farm at the time. Now it's called Janelia. We dropped the farm name because everyone thought we were like a.
This is Bob Dylan song. It's like I know exactly. It kind of pulls. I think it's a perfect job for you because you have this hippie connection. Janelia Farm.
Yeah.
It's like a scientific commune.
Yeah.
Almost. And
when I told my wife about this, there's this place called Janelia Farm, and her mind, and she's from Oregon too. Her mind, she's like, okay, so it's going to be like science, but then you also have like you grow your own vegetables or something, right? And it's like going to be this like peaceful thing. You do yoga in the afternoons or something, right? So that was her sort of thought when she heard the name. That's part of the reason we changed the name.
I think I like Janelia Farm. I think it's what's that Bob Dylan song? Maggie's Farm.
Yeah.
So I don't know. That's when I think about Janelia Farm, I think of Maggie's Farm.
Yeah.
Okay, so what was how did Janelia get in your radar?
Yeah, they had there were a few calls. I think one interesting thing about the model there was hiring guys straight out of grad school. So there are a few of these white-head fellows, etc. But Janelia, explicitly said we're going to hire lab heads straight out of grad school. So I applied, and I applied cold, right? They recruited a lot of other folks, but I was sort of the dark horse there. Yeah.
Cool. I love it. They just something they didn't expect you.
Yeah. No one. Yeah. I showed up and.
Okay, so Janelia, I think Tejan was president of HHMI at the time. Maybe
actually, Tom Shack was still. Yeah. So and this was one of Tom's brainchilds. So,
okay.
So Tom's still president. It's before Tej. Yeah.
Okay, so Tom Shack, he discovered ribozymes.
Yeah.
Huge fan.
Yeah. Amazing person too.
Yeah.
Oddly, the cool thing about Tom's brother lives 15 minutes from my hippie childhood.
Really?
Yeah, in this little town called Williams.
Sometimes things are meant to be. You know what I mean? There's sometimes like reality kind of is stranger than fiction, and sometimes things like used to the oddities of life, and you realize, well, things like that show up, and you start realizing, okay, this is some sort of order of the universe. So Janelia was always, from my understanding, focused on the gene. That was the core, because Eric Betzig had joined, and Eric has a weirder story.
Yeah. Eric's story is
he worked in like a family business, He had a crazy career.
Yeah.
In Janelia, but
So Janelia is this new thing. The founding director is Jerry Rubin.
Yeah.
Jerry did a lot of cool stuff, figured out how to make transgenic flies, led the public effort to sequence the fruit fly genome. So Jerry's the founding director, and Jerry, has this idea, I guess on his days at MRC LMB, to create this place where we do science differently, right? Where it's all about collaboration. The labs are small, and then it's all internally funded. So that allowed us to pick up sort of we call atypical group leaders at the time. So Eric Betzek was the first leader, and he and Harold Hess had cooked up this super resolution microscope in Harold's living room. So then, Eric was the first group leader at Janelia. And this was after a 10-year hiatus where Eric had worked for his family.
I know. I'll talk to him. He's in Berkeley. He actually, I need to figure out. I need to go visit him. But his story's crazy. It's just like, what? Like, how did you?
He was at Bell Labs, and then he just kind of. You have to know Eric, but Eric, he's not afraid to make big changes.
He has a quote. He said, "Without Luke Lavis, some the equivalent. Without Luke Lavis, he's our secret weapon." Essentially, it's a quote.
Eric and I are good friends. It's.
So, what was your first job at Janelia? Was it like just technician or
No. So I mean, this was early days. There was like one round of hires, and then I show up at this interview for Janelia. I have really no clue what's going on. I walk in with my suitcase, and it's a good friend of mine now, Scott Sternson, another chemist, or at least trained chemist now. He's a neuroscientist currently at UCSD. So Scott's just sort of loitering in the foyer, and then I walk in, and then he comes up and he's like, "Are you Luke Lavis?" And I said, "Yes." He's like, "Let's have a seat." And so you started at that point, I guess. And so we had a 2-hour dinner.
Yeah.
And then you get up next morning. There were 8 of us. We gave 20-minute talks. We had panel interviews. We went to dinner at Jerry's house, and then we flew out the next morning. And then exactly 1 week later, I got a call saying, "We want to offer you a 5-year, at the time, fellow position.
Cool.
Please come." So it was it was like trust your gut type stuff. They did not. This was not a long interview process. This was not a long decision. Yeah.
And did you have like a pitch going in, like, "Hey, this is the problem I want to work on," or was it just more like, "This is what I've done, this is who I am, and where I like"?
It was a bit of both. You had to write a research proposal. I honestly worked on very little of what I proposed, but I can think of 2. I know when I got the job, and there's another thing that I think helped. So during my talk, I presented all this work on making all these different fluorescent probes for studying this problem of protein internalization. And one of the questions I got at the end is, "Hey, we work a lot in vivo, in intact animals. How are these probes going to be useful?"
Mm-hmm.
And the standard chemistry response would have been, "Well, we're going to make the probes, and you guys got to figure this out." And I said, "Yeah, it's a huge problem, and it's something that the chemists have to deal with, right? It's not your problem, it's my problem."
Yeah.
And I know that was the moment where they realized I was going to be collaborative. We were going to work together to solve problems. I wasn't just going to be there making molecules, hoping that someone was going to use them.
Oh, interesting.
Yeah.
Cool.
Yeah, and then the other thing, a good friend of mine now, Josh Studman, asked me during the panel interviews. He's like, "You realize this idea that you proposed probably won't work?" And I said, "Yeah." And I think the whole room got silent, but at least I was honest, right? And that's something we value at Janelia, is being honest and, having constructive criticism of each other's work, right? It's not a bad thing to say, "I think this is a terrible idea," right? And you respect one another.
Why is that? Because I think I have a lot of friends who run labs, and when they make that progression from grad student, postdoc to running a lab, is grant writing, and that is another something. I'm assuming you don't write grants.
No, we don't write grants.
Exactly. So like you avoided the grind of grant writing. Do you think that's a big reason why Janelia has a different culture, or
I think you identified. Yeah, it's you have to. No one else is going to hold us accountable.
Yeah.
Funding agencies aren't, and that is what makes Janelia special, right? We can move fast, we can be nimble, but we do need to make sure we're not fooling ourselves. And so we rely on our colleagues to say, hey, you know, what do you think about this idea? This is dumb. Because I mean, early on, you know, they hired a bunch of folks straight out of grad school and gave us very large budgets and with no oversight, right? And so a lot of, you know, I remember sitting with Josh late at night. Do you think we can be here? And he's like, well, I guess we're lab heads, so we can do whatever we want. And you know, they're still in that kind of grad school moment, you know, grad school mindset. And so, yeah, you know, the problem with that is you can have an idea in the morning, you can order, you know, $20,000 worth of reagents or chemicals or something to try that idea, and then you can actually go to the literature and figure out something that doesn't need to be fixed and it didn't work. Right, that was a problem, and so I think we learned as a group that we have to really discuss these ideas, because we're not going to, you know, that we have to rely on our colleagues to vet our ideas.
So maybe it's the reason why you went to CZI today or present, or it's because just kind of just showing your research and trying to get feedback. And
I think you know, I mean, Janelia we wrestled with a lot of these problems. CZI just started this also new imaging institute, and you know, I think we're going to work in a collaborative fashion with them, making sure that, you know, we're all just going to try and solve this problem about biological imaging. But you know, we've sort of lived through this. We figured out a way to identify important problems and then solve them without wasting time or resources.
Cool. Is there like I've never been to Janelia, actually. I should go visit. I'm gonna come visit him now, and we can go look at the microscopes, and also you should have like something. You should bring like some sort of calming thing to it, like apple picking. You know, like cook your own like potato. Pick your potatoes and cook a potato or something. But
no, I mean, like, so cool. You know, during the recruiting phase, right? My wife and I are there, and we're we're having lunch with another head at Janelia now, Vivek, who had also come straight out of grad school. He you know, a lot of us did something interesting, then went to grad school, and then to Janelia. So he had worked, and then went to Caltech, and then came. And so, you know, Vivek's trying to sell us on this place, right? And you know, he like left to go do something at lunch, and my wife leans over and says, "Is this a cult?" I think so. So yeah, well, it's not a cult, but it sort of felt that those early days of that energy. It's, you know, because like this is kind of too good to be true.
Exactly.
It's like you get to come here, you get anything you want. It's a beautiful campus. It's a cool place, and you can just really do whatever you want.
Okay, I need to, you know, let's all get rich, and I think it's really, getting scientists rich, hopefully make more Janelians. I think that'd be nice to have. And so maybe we can talk about the research you do and the lab you created, the group you created. How did you establish your programs, influenced by other groups and what they needed, or did you have like a set game plan? And then maybe we talk about 2013 paper you published, or 2013 papers you published kind of maybe 5 years apart. One led by Brian, another led by Jonathan, where you just create these libraries of imaging dyes, fluoro series, which is kind of probably the most impactful work you've done so far, in my opinion. Maybe we can talk about the research you do, and then kind of the stories of those papers. So you're Janelia. How did you determine? How did you figure out to work on? Because like you have a sweet gig. Like, 5 years, I can basically do whatever I want. I'm in Virginia, pretty sweet. I'm in like a place I like living. Like, I would be perfect, actually.
No. At the time, it's like, this is a shot, right? I got to do something interesting. Yeah.
Like, I'm just trying to get to, like, because it seemed like even in grad school, you get time to reflect and think about what you actually want to do. And that can be a privilege, but also can be kind of hard because you have all this optionality.
Mm-hmm.
How did you narrow down what to work on?
Mm-hmm.
You had the world's your oyster right now at Janelia. How did you mess up? How did you actually like work on things that are useful?
Mm-hmm. Yeah. So, one of the limitations of Janelia, which is engineered into the system, is the labs are small. So we started out with 2 people. So, I hired a couple people. And then what I decided was, yes, I have all these resources and I can do whatever I want, but I was like the one chemist. I'm still kind of the one chemist. It's terrible if I was here just doing random chemistry and not really being integrated into the larger Janelia community, right? So we decided that every project we would do would be a collaboration. Collaboration is huge at Janelia. It's and it's one of our founding principles. And so initially we were working on imaging probes, but we're also working on neuroscience. So, at the time it was 1/3 imaging, 2/3 systems neuroscience. Now it's evolved a little bit. So I had folks like Eric Betzik, Harold Hess, the super resolution, localization microscopy, Palm, and all those things had come out just a few years before. It was still a really big deal at the time. And then there was all this systems neuroscience stuff. So we had projects along both lines, and I'll admit it was hard to balance those two. So our first few papers were on imaging probes. We also had this paper on targeted molecular delivery, and the idea was to target indicators or dyes or drugs to specific neurons in the brain. So I was trying to balance both of those things with a very small lab, but everything was collaborative.
Okay, that sounds overwhelming. That sounds like a lot of work. Sounds like a lot of stuff on your plate. Sounds like a lot of people. Like, did you seek out the collaborations? I'm trying to just understand. You're the enabler for a lot of people at Janelia and just imaging in general. Do people come to you with ideas and then you have to like curate them, or?
Yeah, I wasn't very good at the beginning. I think the spirit of Janelia is you just, it's what I tell people is I'm willing to try anything 4 to 6 times, right? Because, it's like I don't have to swing for the fences. I can swing for the fences more than once because I have that freedom. Yeah. And, we did crazy stuff, right? Like every once in a while, like I'm cleaning out the lab and I find some relic of that time. I have like this $3,000 high shear mixer and we were trying to get carbon nanotubes into resin. So we would fix a charging problem in EM for Harold Hess, right? And we tried a bunch of stuff. A lot of it didn't work. But what ultimately, we got traction coming back to, grad school and everything was really investing in the chemistry of small molecule fluorescent dyes. Because again, in grad school, I was using this 1887 chemistry to make dyes. At probes, I was using that chemistry, just cooking things in really long acids. That was the way we made rhodamine dyes. Yeah. And, I was slowed down during my PhD because of this, right? I could have probably got more stuff done. And so, I show up and, one of the folks that joined not right at the beginning, but a little bit later was this guy, John Green. Cool. It was someone I had known in graduate school, and he had gone to Merck. And so I eventually convinced him to come, like, hey, make some dyes with me.
Cool.
Yeah.
Maybe we can give a brief primer on probes, imaging dyes, because this is something that's like been going on since the 19th century. Like, maybe the history of dyes at the key moments.
Yeah.
And how do you think in the current era we're in and the work you do, like, what were the big problems when you joined Janelia, and maybe those problems still kind of exist, but maybe like when you think about the history of imaging, especially from imaging dyes, what are the key moments that kind of got us where we're at right now?
Yeah, so this whole field of dyes in general, just meaning color compounds, that are made in a lab. This started in 1856. This guy William Perkin, you know, like kind of goofing off in his parents' attic, accidentally discovers this purple dye moving, and that's like all of a sudden, you know, that turns organic chemistry from this curiosity into an industrial powerhouse. And so a lot of the companies that we still think about today, BASF, come from the subsequent decades where dye chemistry became important. And so they made a ton of dyes, really until the 1930s. Some of the early drugs, for sulfa drug was a dye that they were screened. You know, we get Tylenol because Tylenol was made because there was a byproduct of making dyes. So they're like, oh, we got to figure out how to use this stuff, this byproduct of our dye production. And that's where we get. And so, you know, a lot of the fluorophores that we still use today were discovered during that time. So, you know, there's fluorescence is sort of in the mix. It's not until the 1940s when Kuhns says, hey, we should have more antibodies. And that's immunofluorescence. And then in the 60s and 70s, people figured out you can make lasers using dye solutions. And so a lot of the structures that we still use today to improve photostability or brightness kind of come from the laser dye work. And then in the 90s, we get the Alexafluor dyes and Psi dyes and the Ato dyes and others, right? These commercial optimized antibody labels. Most of that was done for dead cell work, either labeling oligos or antibodies. But in the early aughts, I guess, Kai Johnson developed this snap tag technology. Omega developed this halo tag technology, and this now allowed us to take molecular fluorophores and use them in living cells of animals, right? So little enzyme-based tags that grab onto a little synthetic piece that we can attach to a fluorophore, and so it forms a covalent bond. So you can label proteins inside the cells with synthetic dyes, and then you shine light, glow, and you image.
Makes sense. Yeah. So Luke, you wrote this awesome review on it. Thanks for sending it to me. But we'll link it in this, and that kind of goes to the history to the big opportunities in imaging dyes. It seems like the big leap was then using fluorescence live cells. What happened was that I'm not with all the literature here, but was it around the time you were at Janelia? And what were the kind of implications that, like, hey, we can actually image live cells and then start imaging protein motion, you can image like transcription factors, and begin to like really peer into the cell as it does things rather than looking at like stains and stuff.
Right. Yeah. So we managed to hit things just right. I mean, it's a little serendipity. So we had that's there. They're making amazing microscopes. Then Tom Check resigned. He stepped down from being president of HHMI after 9 years, and so Teach was president, and even before Teach became president, him and Rob Singer and a few others created this transcription imaging consortium at Janelia. The idea was to image transcription factors in live cells. So these people were secondly through. That's where we got Brian English to come down from. He was in Rob's lab and at Einstein, came down. And so we assembled this team, and I was sort of on the periphery. But the idea was, hey, there are these labeling tags. We have these amazing microscopes. Mats Gustafsson was also developing scopes there, sadly, before he passed away. And so, together we sort of built this little imaging ecosystem to watch molecules move around in live cells. And the idea was to look at diffusion. Teach was, Teach is an amazing biochemist and a lot of in vitro stuff.
I think he discovered the first transcription factor. So something to tip your hat on.
Which is something else. Soft spoken too.
He's like he's a master. He just walks very slowly, and you're just like, I first met Tej on a massive screen
Yeah.
because he's always somewhere.
Yeah.
And he's just sitting there, his face kind of like being like it's like it means like meeting somebody on a movie screen, and you're like, oh, nice to meet you, and you're just like, is a world class science. So you created a transcription factor consortia, and you had Jonathan Grimm. We'll also talk about how to build a group and the culture you've set. But Brian English, some real superstars. Was that kind of the catalyst for you to? I mean, what I'm trying to figure out is like, what was the catalyst for you to develop the Fluoro Dyes? Like, what was it? Just kind of a collection of projects, and you started putting these together, or maybe was it this or something else?
Yeah. So let's see. I want to say.
And maybe you can describe what the series of dyes you produced too.
Yeah. So at the time we started getting involved with this group. There was a little bit of a transition. There was some in vitro work that was being done, some, and then moving into live cell single particle tracking, again, watching molecules move around in live cells. And, we had, Tej had a little group of, at Janelia doing the in vitro single molecule imaging things. They had built a nice turf system with Steve Chu. And so that was sort of the initial project. And I joke about this sometimes in talks is, Tej came in and said, okay, we need a red dye attached to this halo tag ligand, the piece that would grab onto the protein. And so we did it. It took like an hour, but I wanted to get into easy, so we waited. Emailed Tej. Tej was busy. He was presenting at HHMI, so I waited a while, and then, like I said, I think we finally got it, Tej, and then we published a paper with them. But that was the beginning, and then we learned, okay, there's a single molecule imaging stuff, but they were still using the dyes from the 1800s to do this. And so John and I thought, oh, we should make a better fluorophore, and we had developed chemistry where we could make any rhodamine dye we would want. And then the question is, well, what do we actually make? And that's, I think, a big difference in the field now is because of our work and many others. In the before times, the question really was, okay, we're boiling things in sulfuric acid. What can we make? What's going to survive those harsh conditions? Now, with all this new chemistry, the question is, what should we make? Because we can make almost anything. And so I was just kind of goofing off with some very simple computational chemistry experiments. And I had this idea: what if we installed a little four-membered ring to the fluorophore? Would that still look like a dye? What would it be? And so I remember, I ran a calculation, came in the morning, looked at the structure, and it looked promising. So I went to John. I said, "Hey, check this out." He's like, "Oh, I'll make it."
Cool.
And that was the beginning. I still work in the lab. That's one of the things Janelia group leaders do. And so I was doing some spectroscopy, and then John had made this dye and he was purifying it. And there was this eureka moment where he pulled the fractions out of this HPLC purification system, and it's like the brightest thing we'd ever seen. We're like, oh, that's going to be a winner. Ended up being a really simple modification. So we really only added 2 carbon atoms to this dye from 1887, but we made it much, much brighter. And that pushed us over some limit to really be able to do useful imaging in live cells, right?
Really, I think the paper got published 2012.
It was 2015.
2015, I'm just trying to remember the timelines. So you published that paper. Did you have a lot of inbound? I'm assuming everybody, how do I get these dyes? So what was you published a paper, and now the world knows about it, and realized, you can have all these dyes across the spectrum. And you can synthesize them, and everybody wants a piece of it. Was it overwhelming inbound, or was it just kind of under the radar?
It was a little under the radar after the paper, but I think in general, chemists tend to publish things and then, we'll wait for commercialization. There's there's not this culture of sharing, and it makes sense because it's time and money. Typically, you have a grad student or a postdoc, and they shouldn't be spending their time necessarily making stuff. They should be doing new stuff and getting on to the next stage of their career. So there's not this culture of sharing. I mean, even in the genetically encoded world with tools that are based on proteins, it's easy to share because you can just share plasmids, and then you have Agene, a fantastic organization that shares things on people's behalf. But so we got some people asking, and then John and I just one day decided, let's just start giving stuff away.
Cool.
And so people would ask, where can I buy this? And I said, we'll just send it.
Cool.
And they're like, really? Yeah. And then we also decided just sign paper. This other thing in chemistry often with the culture is, well, I paid for that synthesis and someone in my lab did it. So we got to be authors on the paper.
Mm-hmm.
And sometimes I got to be a corresponding author because you couldn't have done that work without my molecule. And we decided, let's not do that at all. Let's just say, cite our paper, acknowledge us, and it'll be cool.
I think this is like kind of the epitome of the beginning of the story.
Yeah.
Having hippie parents, living in Oregon, living on, remote cabin, and now, 20 years later, almost, it's just like, now you're publishing this work that has a lot of value, and then bringing your own, like, values and to that, and let's make it open access. Mm-hmm. There's Agene for all these different types of plasmids, and you can just essentially, people will publish a CRISPR paper and make the plasmid accessible through Agene. You propose AgChem, which should probably exist, and you'll probably build it sooner or later. You already have it kind of built, quasi-built. Mm-hmm. Maybe you could talk about that. Like how many dyes you send per week to other labs, and kind of like how's the growth rate been over the last maybe like 7 years?
I mean, it started out just word of mouth.
Yeah.
And then it got. I tend to not. I don't do
marketing.
I don't. Well, I mean, I was giving a lot of talks and stuff, and Tej was too, right? So people would be like, "Hey, just heard Tej. Can we try this thing out?" Right? And then I think people were shocked because they would show up the next day, right? But it didn't really have an automated system. It was just email to me, and so I had at one point I got a new administrative assistant, this awesome person, Anastasia, and my old assistant had been dutiful, like and like 20 emails on Monday, like, okay, here's all the stuff over the weekend, and then I would collect everything, handwrite little index cards, and we would get them all shipped out. And then Anastasia came and said, yeah, we're not doing this.
Yeah.
So she created this whole web portal and streamlined the whole thing. To answer your question about how many dyes we send out, I estimate we send out 100, 200 a week. I actually don't know because I'm out of the loop on purpose. So there's no gatekeeping here. It's something we just do for the community, and I'm actually not involved. We have these awesome technicians, Katie and Sarah, right now, who take care of this. They synthesize things. They put little things in bags. We have an organized freezer, and I don't know what's going on, and that's that allows me to not. I can't put my fingers on any scales or do anything weird like that. Right? So I think that's important.
So have you been to a talk or been to some sort of conference or poster session? And it's a pretty cool research, and then you realize, they actually use the dye. Have you had those moments?
Yeah. A lot of times I go to conferences and people are like, thanks so much, and I'm like, I don't know you. I don't know who you are. I mean, I'm thinking, I just picked it, but.
Johnny Appleseed of imaging and chemistry. We're just like, here, just giving dyes away. The legend of Luke Lavis grows by the year. And also Jonathan Grimm and Brian English and a bunch of all the great people. And maybe we can transition towards group culture.
Mhm.
I think Janelia is an outlier. I think
Yeah.
the whole institution has a great culture. But for your group in particular, how many people is there? I think 6 or
Yeah. So
Yeah.
we start with 2 people at
Yeah.
Janelia, and the largest lab is 6.
Okay, so then how did you think about what kind of lessons you've been in Janelia for
15 years.
15 years? Wow,
Speak, yeah.
that's congratulations. You have kids during that time or
Yeah.
family?
So yeah, 2.
Were they born in Janelia? Were
Yeah, they were born in Janelia. Yeah. So
True
yeah.
family.
Yeah. Eric, tends to joke that the biggest impact Janelia has had is the number of children. There have been a lot of kids. Yeah.
Cool.
So yeah, my kids. So my son Linus. So we named him after Linus Pauling, another Oregonian chemist.
I think
and
Linus
daughter
has the
Clara.
same saying where you have this, you're saying, I'm willing to try things 4 to 6 times.
Yeah.
Linus has his own little
Yeah.
number of ideas. It doesn't matter how many ideas.
Yeah, you need a lot of ideas and a good selection process.
Something like that, exactly.
Yeah. No, Linus Pauling, another Oregon State grad, actually.
Really?
Yeah.
Oh,
So,
I didn't know that.
Yeah.
Cool.
Pauling, amazing person, and also a little bit of a maverick, right?
He was out there.
Yeah.
He won the Nobel Peace Prize and I think chemistry Nobel Prize, and he that very famous image of him proposing the triple helix.
Yeah.
This is it.
Yeah.
And then it was true, but
He swung for the fences a lot.
you know what I mean? Yeah, he definitely did. And then okay, so you did that for 15 years. What kind of lessons have you learned about leading a scientific group,
Mhm.
recruiting people, retaining them, and then trying to set a culture of you kind of alluded to this, being useful versus being right or something else, or being like, how do you over the last 15 years, what kind of lessons have you kind of learned about culture and leadership?
Mhm.
Especially for scientists, because scientists are kind of a hard group to these times versus you're in a company everything aligned with money or product, but science is it's jazz almost. How do you lead a jazz band of scientists?
It's true, right? Everyone is unique.
Yeah.
And I think one interesting thing about science, which Hughes and others are hopefully going to help solve, is you're a good scientist and you're chosen to be a PI because you have good hands at the bench and good ideas, and then they place you in essentially a small startup biotech role as CEO, right? And so you have to learn all these management skills kind of on the fly. So the important thing is to have good mentors.
Yeah.
I had fantastic mentors, Charles Zucker out of Columbia, and also my colleague Scott Sternson, the guy that met me and grilled me in my first day at Janelia. Scott and Lauren Luger, also at UCSD now, who's just at this workshop, so it's good to see him.
Cool.
Have good mentors who can help talk through things, because you're not going to know what you need to know starting out running a lab. And then the other thing that I learned is, not everyone is like me. I benefited from a very hands-off management style. I could figure things out on my own. I enjoyed, just kind of coming in and doing things. Not everyone's like that. People need some people high touch. They need a lot of, they need to talk things through, and so you have to figure out a specific management strategy for every single person in your lab. And that's okay. Yeah,
I guess it's possible for a smaller group. So maybe you also that's kind of a feature of having small groups of Janelia versus 100 people labs. And,
if I had a 100-person lab, I could do this, but I could have personalized mentorship style for every single person.
So it seems that for a smaller group to be as productive as you are, you really have to find the right people for the problem and the work.
Yeah.
And you've recruited some superstars.
Mm-hmm.
It seems from talking to you, it came from your network. Oh, this person I knew from grad school, come join me. I knew this person, from Ruben's lab, come join us. How do you think about, how do you cultivate a good taste in people, especially to work with
Mhm.
on research. Because like that seems like, how do you like it seems like the group you run is like special forces and versus standard army.
Mhm.
And how did you get good at like filtering, making your group just really cohesive? And
Mhm.
it seems to be a challenge for a lot of people.
Yeah, I think one lesson I learned, or this has been before, like I was kind of worked as a janitor like, from 12 on.
Really?
You've got yeah, this is
That's
like
cool.
another funny story. But my boss at the time, this guy Todd Metter, what Todd taught me was always be willing to do whatever you ask someone else to do. And Todd, Todd, is a construction guy, like kind of legendary. Like you ask someone to do something, and three guys said, "Oh, three guys couldn't do this." And he went up there and did it all himself. It's like one of these people. But it was a lesson I learned early on. And so, it's about treating people with respect, and also being willing to do whatever. If you're going to ask someone to do something, it should be worth your time, not just their time.
I bet it's pretty inspiring that the people in your group see you in the lab as well.
Yeah,
and like getting your hands dirty and like actually like
not as much. If they're listening, it's not as much as I'd like, and not as much as I used to. But I do get in there from time to time.
Yeah, that's useful. Cool.
Yeah, and then I think the other thing is trust your gut.
Yeah.
Another important mentor is Jerry Rubin, the founding director of Janelia, and what he always says is, your gut's going to be right 85% of the time. So you have a choice. You can either learn to live with the 15% wrong decision, or spend the rest of your time trying to get to 90% right decisions. And so he's like, learn to live with the 15% failure rate, and then you'll be fine. So it hasn't been, like my decisions in general haven't been all perfect, but it's about this person gets it. You can usually tell just through a brief conversation. This person understands, is passionate about science, understands the problem, understands what we're trying to do.
Cool. Awesome. We can talk about that lastly, maybe penultimately. Like, what are the big problems we're working on now for imaging? What are the big problems in imaging probes? Is it trying to make them more photostable, brighter? Is it trying to find applications in neuroscience and trying to like find like biosensors and live animals? What kind of things are you working on now?
Yeah, so one thing I can list a bunch of different things. One thing we're really trying to do is democratize resolution microscopy.
Cool.
So that this is a technique that came out 17 years ago now, and it's still not that easy to do. And so we're trying to make super easy to use probes and then also give them away for free.
Cool.
So that's one thing, even, we want to know anyone can use this stuff, not just an expert, and hopefully not having to invest a lot of time to get some photoactivatable thing to work, right? So we're working on that. I think the future of all biology is going to be in vivo studies. So we're moving from cells to tissue, ultimately to in vivo work. So everything we do in a cell culture system now, in 10 years, we'll probably do in vivo. So for the field I'm in, small molecule fluorescent dyes, we have to figure out how those dyes behave in an intact animal. And we have to treat dyes like drugs. And that's the thing we're not used to as a field. And it's expensive. We get something to work, we get a cell to glow, we get a paper, and that's it. So we've invested a lot of time in figuring out the rules around bioavailability of a lot of our dyes. And we now have a suite of fluorophores that work in vivo, and that allows you to do cool experiments like pulse-based labeling. And then the other thing that we are really thinking about, and even this workshop at CZI, we were discussing, we've been making fluorophores 50 years now. We don't know the chemistry behind a process called photobleaching. You illuminate them over time, and then eventually there's some photochemical process that destroys the fluorophore that makes it not fluorescent anymore. We actually don't have a clue what's going on there. No one's ever investigated it. So it's boring work. Probably would have a hard time getting an NIH grant for this, but it's something Janelia can do. That's something we're going to really invest in the future.
Cool. Okay, so look back at your really very career. You've been exposed to so much, and it all seems intertwined with growing up in Oregon and just being lucky at probes too, where it seems like so from outside looking in, so diverse.
Mm-hmm.
But then in reality, when you hear it, you realize, oh, it kind of starts from this one small decision, and it built up from there. Kind of any lessons you've learned in terms of choosing a career, figuring out who to work with, especially for scientists, a lot of PhDs into this who are trying to make their own decisions. Kind of any takeaways so far?
Yeah, I remember, I had this decision to make. Would I leave? I still love Oregon. Everyone knows I love Oregon, and I had this opportunity to move down here to work at molecular devices.
Yeah.
And I remember sitting with my then girlfriend, now wife, coffee shop, and remember she said, "Let's just do it." Right.
Mm.
And actually everything in my life is because my wife prodded me into the concrete layer in Southern Oregon. If it wasn't for her, but taking risks is important. And sometimes, you don't think you can do it. It's scary. It's like first generation college student kid, worked in this safe environment at molecular probes in a familiar place. And then suddenly moving down to Silicon Valley, it's a crazy time. But that was worth it. And it was a risk to not stay here, to go to Madison. Why would anyone go to Madison, Wisconsin? And then again, taking a risk to go to this new institute in Northern Virginia, kind of in the middle of suburbia, that was untested. But I think taking risks is important. You don't want to be safe, especially when you're early career, right? You never regret not taking a risk.
Yeah.
I'm sorry, you never regret taking a risk.
Yeah. Just like having a good support network.
Yeah.
And then just going for it
Mm-hmm.
when you don't have abortion kids and stuff like that.
Yeah.
It's how do you something I have struggles with. How do you deal with transition periods? I have a big struggle when I move somewhere else.
Mm-hmm.
It's like a 3 to 6 month period where I'm just not established. And how did you move from probes, devices, Wisconsin, Janelia, even Janelia for 15 years? And I'm assuming when you're in your groove, probably have a routine, and there's some advantages to that. But in those early days, how did you feel unmoored?
Mm-hmm.
And still be productive, because it's hard to explore and still do things.
Yeah, well, something my wife and I talk about a lot is having lived in many different places now. You realize that no place is perfect, and so the goal is you maximize the good things, and then you minimize the bad. Right, there are good and bad things. Oregon is amazing.
Yeah.
There's forest fires and it rains a lot, right? Virginia, the winters are a little bit rougher. The summers are humid, but you figure out what you can do. I live very close to the Potomac River, so I paddleboard every day in the summer, and it's like you find joy you find the special thing about a place, and you invest in that, do that, because it's the old saying, you can't go home again. Sure. If I ever go back to Oregon, I'll be like, there's good and bad things here too, right? I realize it. I think it's fine, but there are great things about living in Silicon Valley, great things about living in Wisconsin, and bad things too. But we cross country skied in Madison and did a bunch of biking. Down here, we did all the cultural stuff and went to the ocean whenever we wanted. it's minimizing the bad thing and maximizing the good.
Cool. Okay, take risks, family. Anything else? And you have some notes. I'm
Yeah.
very curious about your notes. You wrote a lot of notes, Luke. How many notes did you write? You wrote a lot. Anything else, or?
No, I think I'm the one thing I wanted to say is, it's. I think we're entering a new phase in science where generosity and kindness is valued again. I think the cutthroat, competitive environment is kind of going away because we realize we have to collaborate. It's really rare for one lab to be able to do all these things well and really dominate a field. And so, working together is going to be a better way. And it's okay to be generous, be kind, be honest. Sometimes you get a little screwed over, but you get screwed over more if you're like a competitive jerk. So it's totally okay to be a nice person and do excellent science.
That's awesome. There's the Beatles song, right? Love you take is the love you make, or maybe I always get it confused that like at the end or something, where you just want to put more stuff out there, and then you'll get some of it back. You probably a lot of it back, and so. Cool. I mean, thanks for doing this.
Yeah, no,
You're sort of visiting here, and this is probably the best office in Silicon Valley.
I know.
We should go get some snacks right now, and then get you. Are you fine now, or?
Fly back tomorrow.
Okay.
So,
I don't want to make it.
Oh yeah.
We're not flying. I know you're flying out later today.
Oh no. I mean,
I feel a little bad. I just want to get something to drink. Because I'm like, you've been talking. I'm like, take a break, or I'm sure to keep going. But thanks for doing this. I really appreciate it.
Fantastic. Yeah. Thanks for having me.